And then and you wanted to go over the first exam, right? Whitney? Oh, problem set questions.
Yeah.
Oh, shoot. Okay. If they're over like some of the last problem sets, I will just have to freestyle them because I didn't do those because I was just like focused on the practice exams. But I will have to attempt them.
I think it's any VP. Yeah, it's making the barriers and it crosses is reversal. Which makes sense, but they do.
Okay. So what are the questions we got?
I know in the session we said that like the 3 regions of LPS are more similar to those specific antigens. I did not write why.
So the 3 regions are, say that again?
The 3 regions of LPS are capsules and they're more similar to the O specific. And why is that?
Can you see the question?
Yeah.
Oh, just slime layers.
Yeah.
So slime layers are, if I remember, like the most variable, or I mean, they're just okay, so what we know about the O specific area is that it's just like a long chain of variable like carbs. And then if we go to slime layer, I'm sure it'll say something similar to that.
Oh, it just says in the slide.
Yeah, so it's made up of long polysaccharides, and it literally says the answer itself, like the O specific antigen. Yeah. What else we got? And 1 for 0.
You have your answers for the first practice exam?
I do. I actually have someone that came to my Tuesday session that wanted to go over the first one.
Okay.
So I want her to come in first. Yeah. But she said she'd come 15 minutes early. Oh. So what state are they later? Oh, state. Well, if they can't do respiration, that's essentially, I think that means like, it's just, it can't do that end step where like you pump hydrogens through the ATP synthase. I don't know if that means that they're not being pumped out.
I didn't know if it was like oxidized or reduced. Like if that's what it was trying to get at.
So like if you're not pumping things out, like you're not going down that redox tower and you're not pumping like hydrogens out into like the extra or the periplasm or whatever, that means that all of your electron carriers, NADH, FADH2, will stay in their reduced state.
Yeah.
Meaning that they have they'll be NADH and FADH2. Like they won't lose that hydrogen and then move those across the membrane. So that's what I think the answer would be.
So they're in their reduced state.
So reduce is
NADH.
you have electrons and hydrogens, but you don't have oxygen. Yeah, so they'd be reduced.
We are for like 30 minutes.
I always mix it up, especially like after NCAT stuff. I just dumped all of that shit. But I think they would be in the reduced state, right?
For respiration and fermentation.
So fermentation, if it can't do fermentation, right? Is that what it's asking? So if it can't do fermentation, that means that essentially the only reason you would want to do fermentation is because you want to cycle things back into the start of glycolysis, right? If you can't do that, obviously like you won't have that NADH or you won't have the NAD pluses, right? Like if you're just going through glycolysis to begin with, if you're just like going through glycolysis and then turning the NAD pluses into NADH for like citric acid cycle, that's running fine. You'll be producing those NADHs, but you won't be shuffling NAD pluses back into glycolysis. So everything should be NADH, which is also reduced state. That's what you were getting because you were nodding, right?
Yeah, we were arguing about it, and I was telling her I was right.
Okay, cool. It's not a competition,
It
come on.
wasn't. We're
It's a
just
collaboration,
here.
right? Cool, what else do we have? Okay, I'm gonna go grab some water. Be right back.
It's also recording, so don't touch it.
Wait, so is it a reduced state? Okay. And this one or should we do this one? Okay, that's fine. And is it this one? Is it this? Okay, I don't know which one. Is it in the bottom? Yeah, but it's in the body. We can.
Good vibes, yeah, we bottom. We started off from the bottom and we still here. Stipends got me solemn. With 2 weeks and $150 worth. I guess I'll get a job. I guess I'll show up for my moms and my pops approval that I am an average guy.
Yeah.
Hey, welcome in.
Wasting my time, just roll up.
Hey, if you haven't signed in yet,
catch
go
up on
on.
sleep and we spent the whole semester up in
Do you
the
wait?
deep
Are you
end.
second time or so?
No, just first.
Okay. Do you know why? Do you know what happens if your attendance is bad?
Smoke
like-
shit, take my mellow.
I don't think they do anything.
My lucky charm.
But I want to see the numbers
marshmallows.
Yeah,
for my ego.
Judge
Not
by
for
the color.
I
Nobody knows green or meadow.
I had to work the first session.
I don't got much,
And,
but I roll with it.
Sunday and Monday.
All my good friends, they roll with me.
Because we just had it.
If you got a problem, take it up with me. Just talk about
After
how my
after y'all's
back
exams,
got roll. If your boys got beef, we
And
can
my
roll
mom
with it. In the club or the street, we
Okay,
can roll.
I had to
with it.
ask
It
my-
don't make me done, blow,
said-
blow. with it. Crack his head with a gun, I'm a soul.
After
Got
that,
weed,
my mom's like,
dudes got
"Well, we're
bills,
gonna start right
dudes
now."
got their work on the corner.
No, nobody's gonna want to do that.
You haters got shit
So,
in
what
they
do you
brain.
No, exactly.
Like,
Got some game kids.
I was thinking I'll just do like an open
No
question.
feds, got the feds on the fence.
Like if you want to come in and ask like,
Job, sitting on the fence. Fall
I
out
don't
in
know.
the club with our kids staying trip. We never wrote a check, just the big face bills. Where I play it, drinking makeup, vodka, cranberry vodka. When I'm
oh,
that's
I'm gonna
right.
do that. Oh,
See a big girl and the player had to stop her. The name wasn't Silk, but her face was the shocker. Fans taking pictures of us falling, but I got him. 7 foot a hole for his body, we gon' drop him. Always on the grind, we be watching when they watch it. And when they turn it back, it's that clicker, rock 'em, yeah. I don't got much, but I roll with it. And all my good friends, they roll with me. And if you got a problem, take it up with me. Let's talk about how my back, I can't roll with you. Boys got beef, we can roll with it. In the club or the street, we can roll with it. It don't make me none, blow for blow with it. Crack his head with a gun on the show.
1, 2, 3.
yeah, you can say I'm responding to another question. oxidized form is the one with more oxygen or hydrogen. Like, that's gonna want to take it. Because once you're in the reduced form, it's not gonna want to get further reduced. So I think that's what it means by that. Come on, bro. 3 for 0. Let's go. What's up? Great. How are you? Thanks for coming. Miami games? Anyone else going to Miami games? What seats? What section do y'all get? You, man. Dude, it's like right behind end zone, right? Wait, where'd y'all go? So I got ticket full around 5:30, and we only got 2:33. But like it's at the 40-yard line, so I'm like, I'd rather take that than the end zone. No, yeah, absolutely. Yeah. If it was a day game, you cashed out. That's like under the second deck, right? Yeah. But I think I'm going to like Northgate or something on Friday, but. The fuck would I go to Dixie Tree? I mean, usually like, I really like the pickle shots. So like, Tipsy Turtle. There's a really cheap one. I think dry beans cheap, right? It's relatively cheap. I just follow other people.
Yeah, I follow other people. We always end up at the backyard for, I don't know, some reason. I know the backyard. It's always like
rolling through the city.
it's a great place.
I like the
I
Riding with the top off.
I want to go there when there's
Man, my
live
whip
music.
so
I
big
feel like
when
that'd
you
be
hit
really
it.
Go Friday into the
Fuck
backyard.
around and get lost. Tell my bitch to let
No, I think I might say like, oh, I have COVID, can't go.
What the
Sorry.
shit
But I'll see you
called?
at the game. No, no, I just don't
Tell
want
me,
to
baby, if
go.
you found that life right now.
I want to sit in.
So,
Oh, what the hell? You gonna sit in your potato chips?
I could put you
Yeah, play 2K. That's it.
What's up?
You gotta go outside. Bro.
Let's take a trip.
No.
Come on,
Have you
that's
ever read the
go
world
outside.
is yours
Come across something different.
on a blimp?
No, I think, I don't know.
Tell her be free,
I'll
baby,
figure it
spread your wings, get your legs in the sky like a plane. Let me guide that. I'm the pilot.
I don't know what I meant that.
Can't nobody see you 30,000 feet on your knees in the products make you freaky shit come up out of. Get high, baby, roll one. Cloud nine about to go up.
The last part is classic.
Loving the feeling, the turbulence, get when we turn
Have you
up.
taken an exam yet?
When we land, we can roll
That's
out.
why I'm.
Show you something
Dude,
you ain't know about.
the lab
Tonight we be
exams.
taking off like with the camera to show out.
So don't.
I got you in
don't
the
get
air,
coffee.
your body in the
Coffee
air.
too early.
How I feel up here.
Hey, welcome in. Could you sign in when you get the chance?
You can
No
scream
rush.
as loud as you want, loud as you can. Ain't nobody gon' hear. Would you like it better if you hit the West Coast? Tell me if you found that life right now.
Hey, do y'all have like a class
Hope it ain't
study
tough.
question document that
I
y'all
could
work
put you in
on?
Mile High Club.
Is it good?
What's
Like,
up?
do people answer like?
Let's take a trip.
Yeah.
Have you ever read the world is yours on a blimp?
I would be careful with that.
Tell her be free, baby, spread your wings, get your legs in the sky.
My friend sent me something and I'm like, isn't this the oxidized form or something?
Can't
But it
nobody
says
see you
reduced or
30,000
something.
feet.
No, it says oxidized, but it's reduced. And that was the same thing when
Get high,
I took
baby,
the
roll
class.
on.
I was like looking at the
Cloud nine about to
document,
go
I'm like,
up. Loving the feeling, the
somebody
turbulence,
was
get when
really
we
short on
turn
time
up.
with their essays.
When we land, we can roll out. Show you something
If
you
you
ain't know
have to
about.
use it. If not,
Tonight
it's
we be
always better.
taking off like with the camera to show out.
I got it. Listen, you need a nigga that's gonna
It's
come over
6:51,
and dig you out.
and the person
You need a
that
nigga
wanted
that
to
you know
review
it's
it
not
is
gonna
not
run
here.
his
So let's go over the first
You
one.
need a nigga
Before we
when
start.
he
How about
done
that?
probably gon' put you out. You need a nigga that's gon' put it in your mouth. dick so big it's
Oh,
like
should we
a
just
foot
run
is
through
in
it?
your
Okay, cool.
mouth. And he ain't babysitting my
Shoot,
kids
where's my
out on your
answer key?
couch. Oh, you nasty. Oh, you nasty.
So we'll do it this
Both
way.
graduated, so fuck, keep
I think I
it
remember the answers
classy.
anyways.
They love me in the shot like MJ. They love me in the shot.
Oh,
like
actually,
no
one
nigga
more
can
thing.
block, not even the
I brought candy.
Compare the code with yourself in it. So
I brought Chinese
so
candies.
My new
They're like
shit so
milk
fire,
candies.
nigga, check my
So if you
profile.
answer the question-
Who you know make waves in a low tide? Debo, your bitch, now she
Some
both
people
eyes.
may have been
Nigga,
you were
real
here
brown
for the
again, my
Tuesday
liver.
one, right?
Pretty brown thing in my bed. Been a long time since I had to ask for head, so goddamn don't make me big.
So first question,
For real, girl, I need you.
read it out and then answer it.
Take off the night.
Oh, for a piece of candy. Go ahead.
It would be,
Read it out, man. Come on.
okay, what would happen? What would happen if you perform a gram stain on a gram positive bacteria, but you forgot to add iodine?
Mhm.
So the iodine is the one that binds the purple. It won't be purple, so it would be pink, right? Because of saffron. in it. So,
Yeah.
Alright.
Does anyone disagree with that? Anyone get anything different? Because I would love to prove you wrong. No. Okay. Okay, next question. Okay, what is what of the following? Sorry. Which of the following bacterial phyla is characterized by high G and C bases in its genomic DNA? Very cut and dry. I'll tell you that the second exam was harder. I kind of put more effort into it, but it's actually going to be harder than the actual exam, in my opinion. So it's going to be good practice, obviously. But someone just yell at the answer real quick. Yeah, there you go. Cool. Question 3. What is the term for the area around a streptomyces colony that kills neighboring microbes to protect itself and its food and all those extracellular enzymes that break down those macromolecules? What is that called? We gotta finish this in 5 minutes. Zone of inhibition. Here you go. Y'all both answered it, so I need to kind of ration these though, if I'm running out. What would happen if you destroyed a bacterium cell wall and dropped it into a hypotonic solution? I'm gonna answer that real quick. I think it was a positive verb. So why is it not B or D? Well, since we already answered A, why is it not B?
Because the cell walls, right?
And what's the primary purpose of the cell wall? No. Osmotic pressure, yeah. It's to protect against osmotic pressure. Secondarily, it maintains shape. I'm gonna pull up the answer key because I want to make sure I don't. Please don't pull up on the main thing. Cool. What area of the LPS is characterized by variable lengths of repeating carbohydrates? Wait, who answered the last one again? Oh! Sorry. Not the I'll just say it. Very nice. Oh, can okay, yeah, she'll take it. You don't like it?
No, already got
Okay.
What was that one again?
O specific polysaccharide, O specific antigen, any of those. What bacterial structures are formed when a cell needs buoyancy? Good. Yeah. How are prokaryotic flagella powered?
Oh, so 60 gas.
Yeah.
Oh, okay. I didn't even know something like that existed.
So why'd you answer it?
I'm not like the right one.
Okay, if you know, don't just yell it out. Come on. Go on. All right. How are prokaryotic flagella powered? It is TMF and the periplasm. Did anyone else get different? No? Cool. What is the name of the pathway that happens after glycolysis when the body, well, the cell needs to regenerate NAD+? Maybe not. So citric acid cycle is whenever after you have pyruvate oxidation, it turns it into acetyl-CoA, and then you citric acid cycle is just like continually harvesting electrons from that acetyl-CoA high energy molecule to get electron carriers for the electron transport chain. Whenever you need to regenerate NAD in the absence of oxygen, what? Fermentation. Absolutely. Where is cytochrome C found? Between 2 and 3? Is that what you all said? Cool. Did anyone have questions on these?
Is it 2 and 3?
Oh, wait, no, it's 3 and 4.
3 and 4?
yeah.
Sorry, I'm autopilot right now. It's between 3 and 4. All right, so anyone want to answer this one for us? Because if not, I'm not going to answer it. Someone needs to answer it first. The SI way, right? You'll take it?
Oh.
Yeah, go ahead. It's really not that high pressure. Well, you're right about the proton motive force being destroyed, but that's because the OH, like whatever generic base that we use, they'll just attack the hydrogens, form H2O, and that'll kind of just neutralize the electrochemical gradient. And we know that like the main hydrogen transport that we talked about is like the simple transport. So that one is the most effective transport method. Welcome in. Could y'all sign in if you haven't already? And we'll continue. So the second question is describe how organisms utilize energy-rich molecules such as glucose to fuel their metabolic process. Whenever I went over this one on Tuesday, people were confused. You have the same phone case as me. Sorry. Anyways, it's a very broad question. So like people decided to focus on glycolysis or something, or maybe like Krebs cycle. Just give me your most entire cell, entire body answer that you can. Does anyone want to take a stab at the second to respond? No.
How would it utilize?
So, okay. So, I mean, I guess I can answer it because it's kind of a shitty question. But essentially, whenever an organism takes in that energy-rich molecule, or I don't really want to call it an energy-rich molecule because that's typically what we call like phosphoenolpyruvate or like acetyl-CoA or something, but just like a biomolecule you would get from eating like Chick-fil-A or something. So essentially, when you take that glucose in, you're just channeling it through all of your different metabolic pathways, such as glycolysis, pyruvate oxidation, citric acid cycle. And essentially, you're just continually oxidizing this carbon structure, harvesting energy from it. You can come on in this way. Yeah, take a seat, sign in if you can. And you're essentially just like stripping it of electrons, stripping it of all of like the backbone carbons and stuff like that, creating free energy from it, and then using that to produce energy in the form of ATP with like substrate level phosphorylation in glycolysis. You also have like transferring those electrons to mobile electron carriers that are funneled into the electron transport chain that generates PMF, stuff like that. So it's a very large, broad concept that was not worded too well. But that's essentially like how we make energy from these macromolecules. And if you're joining in late, well, not really late, on time, we're just going over the first exam. If you got here just now and you have a question on any of these, now is the time to ask because we're going to do the second exam. So. Welcome in though. How are y'all doing? Thursday night before the exam. Cool. So if we don't have any questions on that, do y'all know how to get to my Google Drive? No? Okay, so however you've gotten one of my announcements, either through Canvas inbox or email, go to like one of the announcements that has my website on it. Go to the website, and there should be a button that says Google Drive. And then once you're in the Google Drive, go to exam 1 materials, and the practice exam number 2 will be in there. We're all going to do practice exam 2 together. in groups, and then we're gonna review. And let's aim for groups of 4. If you really want to do it alone, you can, but I think groups would be more fun, and it won't be that quiet either. And I'll do it if you're having problems. So get to my website. Once you're here, it'll look like this. Oh, worksheet and materials, that's where it'll be. Exam 1 materials, second exam, and this is what we'll be doing together. And we all separate into groups of 4 now. Cool. Three to 5 work as well. If you want to work alone, it's whatever. Just get into a group. Don't make it weird.
Yeah.
30, baby, won't you meet me by the beat? All these baby ladies, you know what it is whenever I'm busy. When you see it, throwing me kisses, no. 30 degrees, way too cold for pony time. Will I see you at the show tonight? Will I see you at the show tonight?
Hey. Truck to the plane, to the truck to the hotel lobby. Me, I go through underground garages, presidential suite on deposit, elevator up to the room, shower up, and then we hit the club. Touchdown, gotta see what's up. Area code in my phone. What numbers do I still have? Who do I know from the past? Hit one, she say she got a man. Hit another one, it goes green. Must have changed phones on the team. Remember when you let me in between? That was 2017. All good, chuck a look around, find one to see my type. That my dog, and he know what I like. He done found me plenty in my life. Problem is, I meet a girl tonight, then I go and treat her too nice. Galleria credit card swipes. I don't even know if she a wife, but I do know one thing, no. Women, they come, they go. Saturday through Sunday, Monday through Sunday, yo. Baby, I'll love you one day. Baby, we'll someday grow. Till then, I sit my drunk ass on that runway, on this one way.
30, baby, won't you meet me by the beat? Early, maybe later, you can show me things. You know what it is whenever I'm busy. Windy city, blowing me kisses, no. 30 degrees, way too cold for homie time. Will I see you at the show tonight? Will I see you at the show tonight?
Jury in a safe on arrival, pistol on Jay for survival. A lot of people gunning for the title, a lot of people trying to take us viral. I'm just trying to have a nice night. The waiter brought a check for the meal. She said, babe, I think that was a typo. Nah, girl, I got it. That's like, though, I can show you bigger things than that. It's gonna be a minute till I'm back. Let me get your images that last. Amex had a limit in the past. I had plenty women in the past. I done plenty sinning in the past. You don't need to know, but you can ask. Sticking on your backstage pass, all access, cause you bad. You could go wherever with that ass. Show to the shout, to the truck to the club for the party. You go in first with your girls, cause I don't know who's out there watching. But I do know one thing, though. Women, they come, they go. Saturday through Sunday, Monday, Sunday, yo. Baby, I'll love you one day. Baby, we'll someday grow. Till then, I sit my drunk ass on that runway, on this one way.
30, baby, won't you meet me by the beat? Early, maybe later, you could show me things. You know what it is whenever I visit. When you see it, throwing me kisses, no. 30 degrees, way too cold, only time. Will I see you at the show tonight? Will I see you at the show tonight?
You're right, you're right.
Oh man, bitches got my fucking problem. Yeah, I like the fucking got a fucking problem. Oh man, bitches got my fucking problem. Yeah, I like the fucking got a fucking problem. Oh man, bitches got my fucking problem. Yeah, I like the fucking got a fucking problem.
If finding somebody real is a fucking problem, bring them girls to the crib, make it solve. Hold up, it's the taking hella long. Give it to me now. Make that thing pop like it's in me over now. Ooh, baby, like you draw with the shimmy kind. Exact, yeah, like me. Never met a motherfucker fresh like me. All these motherfuckers wanna dress like me, but the chrome to your dome make you sweat like me. Cause I'm the nigga, like how you figure getting figures and fucking bitches. She rolling switches for her bitches. I put my niggas, they getting put up off the liquor. She know my licorice, they say funny make a nigga act rich. But at least a nigga rich. I be fucking drugs like I be fucking bored. Turn a dyke bitch out, have a fucking pause. Peace.
Oh man, bitches got my fucking problem. Yeah, I like the fucking got a fucking problem. Oh man, bitches got my fucking problem. Yeah, I like the fucking got a fucking problem. Oh man, bitches got my fucking problem. Yeah, I like the fucking got a fucking problem.
Finding somebody real is a fucking problem. Bring them girls to the crib, maybe we can solve. Oh, I know you love it when this beat is on. Make you think about all of the niggas you been leading on. Make me think about all of the rappers I've been beating on. Got a feeling that's the same dudes that we speaking on. Oh, word, ain't heard my album, who you sleeping on? You should print the lyrics out and have a fucking read along. Ain't a fucking sing along, unless you brought the weed along. And just, and just drop down and get your eagle on. Or we can stare up at the stars and put the Beatles on. All the shit you talking about is not up for discussion. I will pay to make it bigger. I don't keep no reduction if it's coming from a nigga. I don't know that I don't trust it. If you coming from my head, then motherfucker get the busting. Yes, Lord, I don't really say this often, but this long dick nigga ain't for the long talking, not beast.
Oh man, bitches got my fucking problem. Yeah, I like the fucking got a fucking problem. Oh man, bitches got my fucking problem. Yeah, I like the fucking got a fucking problem. Oh man, bitches got my fucking problem. Yeah, I like the fucking got a fucking problem.
If finding somebody real is a fucking problem, bring them girls to the crib, maybe we can solve it.
Yeah, hold this the finale. My pep talk turned into a pep rally. Say she from the hood, but she live inside the valley now. Vacate in Atlanta, then she going back to Cali. Got your girl on my line, world on my line. The irony, I fuck 'em at the same damn time. She eyeing me like a nigga don't exist. Girl, I know you want that dick. Girl, I'm Kendrick Lamar, aka business to me just they car. That mean your friends is be up too far. See my standards are pampered by threesomes tomorrow. Kill me all dead bodies to the hallway. Don't get involved, listen what the Christian Paul say. Holly, Harry, hallelujah, holla back, hallelujah. Peace.
Oh man, bitches got my fucking problem. Yeah, I like the fucking got a fucking problem. Oh man, bitches got my fucking problem. Yeah, I like the fucking got a fucking problem. Oh man, bitches got my fucking problem. Yeah, I like the fucking
Yeah, but I guess I got it. Do you have a question about it?
Let me get on it. I didn't realize that it started early.
No, I like if you don't know about it, because a few people asked about it earlier. There was no way you would have known. Okay. So we can just go over it real quick.
Oh, you should.
I didn't change my number. I only changed who I replied to. Laura said I should be nicer, but not to you. I love how you were mad at me too. Should've good, but you would think I was upset. You're upset. Don't take it out on me. I'm out of sympathy. Maybe you should leave before I get in. I didn't change my mind. I only changed who I believe in. You're leaving on the outside. But this can be deceiving. I got away, I go to work. You don't deserve to feel so hurt. You got a lot of fucking nerve I don't deserve. So don't take it out on me. I'm out of sympathy. Maybe you should leave before I get too mean. Take it out on you and your best friend too. I should have left when Drew said you were a bad news.
It's really over. You made a stand. You got me crying. As well as you play. But when my loneliness is I'm gonna find out you.
Set two quick questions. Yeah, I appreciate it. So I saw some that it was all because if you miss the iodine, the purple doesn't stick to it. Yeah, exactly. Okay. That's exactly right. So iodine is like this happens. Yeah, or not this happens. Wash it away. Whatever. They should all wash away from the iodine. And then hypotonic is low solvent concentration, right? Yeah, exactly. So if you put it in a hypotonic solution, basically the solution is at a lower concentration. Yeah. Of whatever. The water was rushed out, right? Yeah. Or rushed into the solvent. Yeah, sorry. Yeah, because there's more solvent inside the cell and the water went. Yeah.
Yeah, she's nice.
So what happens after electrolysis in the body?
Yeah.
The substrate level phosphorylation, especially like in this class, is what I'm talking about. Making the, for example, like ATP,
Yeah.
making that unfavorable reaction, turning ADP plus phosphate again, or phosphate, like unphosphate,
Yeah.
turning that into ATP, that's unfavorable. But you need to couple it with a favorable process, such as step 7 or step 10 of glycolysis, to make ATP. So this isn't necessarily to regenerate energy. On the other side, fermentation. What do you know about
That is what it goes through when you don't have access to oxygen, right?
Exactly right, yeah. So fermentation happens whenever you don't have oxygen, because the only way you can make oxygen energy in the absence of oxygen, obviously you can't get oxygen, unfortunately.
Yeah.
So instead, it shuttles back into glycolysis, because glycolysis makes 4 ATP total, and you add 2. So that would be your main source of energy whenever you don't have oxygen, or you don't have something that resets electrons and electron transmission. So to go back into glycolysis, obviously and you need 2 ATP, but what else do you need? You also need NAD, exactly. So when your body needs to regenerate that's
Fermentation.
or NAD will go back into glycolysis so that it can make
So it can, That's more of a general question.
Yeah.
Do you think when you took the exam, was it more about, what would you weigh, like more about the first 2 weeks or the third week? Like, basically I'm asking, like, how much do you think metabolism is going to be on the exam?
So it was weird because secretly there's another professor that taught mine in second exam. So I don't know if it'll be the same for Garcia, but I think I remember more of. No, I think we even covered different stuff in like
Really? Okay. No worries. Fair enough.
Once for me, once for someone new. I wanna do some things you wanna let me do. Gonna find another you.
Do you have a question about the second exam? Heather.
Remember? Is the height the same?
To get to that concentration, we think people that like the 1-to-1 fraction. What would need to happen to the water?
So when you're talking about like hypotension,
Everybody.
so you go from like the water will go from like concentration, or whatever the like solute.
So when you say like low and high, it's all relative, right? So it's relative to the environment. So it won't necessarily always go to the higher concentration. Well, water will, but like it's more of like a relative. You know what I mean? Like I wouldn't think of it that way. I would think just like, where does the water need to go so that there's 1 water per bottle?
Life.
And then
Life is the only thing we need. They need me to go, but I don't want to leave. Rest in peace, little kids.
Fuck a pigeonhole, I'm a night owl, this a different mode. I'ma have to make a pen of 6 on a pinky toe. Heard you with a shooting guard, just let a nigga know. I would have you courtside, not the middle.
Well, it doesn't use
all good love in a minute though.
I think the idea is that
Cause I'm a timid stove. Plus I'm cooking up ambition on the kitchen stove. Hot start to bubble, see the sun, then she got to go. Whole sound suave, but I can't get me cold. Bro, think he John Wayne. I bought him
everyone stop working.
way they
name, babe.
Right now.
Cone, everybody fake
But lose
now.
my shit.
You can crack the code,
So,
down everything.
y'all ready to go over it
Setting
one more
rose
time?
gold, dread talking to you niggas like I'm J. Cole. I could tell her head good before I even know.
All right, where is my exam? Is this it? This is it. All right, first question. So I want to read it and answer it. For a piece of delicious milk candy. Or not if you don't want. Okay.
I won't, okay.
Okay, go ahead and answer.
If one were to digest, cut up a large bacterial.
Oh, there's a typo there. So I'll be accepting both correct answers. What I meant to put a little curveball. I wanted to say the large ribosomal subunit, but it came off as large bacterial. So there are two correct answers in here. Tell me how you interpreted the question and then what you put.
Mainly through the large. 60 now.
Wait, what?
Small.
Yeah, we said small subunit for.
Yeah, so that's one of the right answers. You want a piece of candy? Yeah. Sure. No hands. Anyways. Okay, sorry. So what's the other answer? So why is it not 522 and 23?
22,
Why is it not C? Well, what's the other thing inside of like the subunits? It's RNA. It's catalytic RNA and what? Proteins. Yeah, I think I've heard it somewhere. And since we're doing a northern blot, that's RNA. If you want to do proteins, that'll be west. Snowdrop. Gotta know the acronym. Anyways, next question. Which of the following is true regarding actinobacteria? Oh my God, it's the answer key. Excuse me. Wait, no, it's not. No, oh, I just had the answer on that one. So there was a little hyphen on what? No, it's not the real. Do you think I got that shit? No. No, why would he give me that? Okay. Anyways, if you have the pre-revised version, please answer it.
Oh, I see it.
It is C. Did anyone else is anyone else a good person and did it correctly and like legitimately? So did you get anything different? What'd you get? No, you read the answer choice wrong. So actinomyces is a different subclass of the larger class actinobacteria. So yeah. No, I'm sorry. Okay. No.
Yeah.
Since you did it correctly. Cool. Third one. What bacterial structure does the hammets resemble? And if you were using your lecture slides, this one would be really easy. Phylli, whatever. Oh, bet through. Okay. Yeah, that's right. Anyone get anything different? What structure is used to anchor the outer membrane to the thin cell wall? Want to know? Okay, cool. What would it be? So obviously I'm hinting that it's gram negative with thin cell wall. What would it be if we were talking about gram positive? Very nice. Cool. Let's continue. What is the name of the metabolic pathway that has an investment and payoff stage? Did anyone get anything different? It's the Embden-Meyra-Arnos.
Never heard that.
Yeah, I never.
No, that's why, because that's like the secondary name that it gave you. You guys got to read the slide.
So that is like,
I said that.
I know, I was like, it's glycolysis, but that's not on there. Interesting.
Yeah. Y'all done. You want? Oh, sorry. You good?
Now we know.
Now you know. Good catch. Did anyone get any of the other metabolic pathway? You're different. You know. Okay, number 6 is which of the following is a similarity between complex 1 and 2 of the electron transport chain? So you said C. Anyone else getting? What'd you get? You got B. Anyone guess A or D? Not guess, get. Okay, so what's your reasoning?
I thought FAD.
I don't have an answer for that.
Sorry.
So FAD had 2 values, 2, it becomes Q8, so I know come 2 makes Q8, so I add 2.
And what was your reasoning for B? You just saw it in the slides. I remembered it that way. Sorry, I don't know why I'm like interrogating my bad. So the only thing wrong with B is that it's a lipid electron. It's lipid based. It's not protein based. C would be the correct answer. He does. He can. He can. He can. It or maybe it was only primarily like Siegley that would do like a little bit of a trick question, but I think I remember that he would throw something similar. Also, I said before y'all started that this one will be a little harder than your actual exam. So if you're like making those small mistakes, obviously like it should be assigned to be a little more careful. Or maybe like go into a tiny bit more detail when you're reviewing or studying. But again, it's gonna be harder than the actual. Anywho, question 7. If an organism can perform both aerobic and anaerobic respiration, what would happen after the organism is transferred from an oxygenated environment to a non-oxygenated one? Taking answers now. So got some C's. Anyone else? No. So on Tuesday when we went over it, somebody said B. And somebody said A as well, but I think they just read it wrong. So why is B wrong?
It doesn't carry on as well.
But like why? No, I mean you're right. But
Because
like,
it produces less ATP.
yeah, it has less reductive potential. Most of like the other non-oxygen electron acceptors don't reduce as much. And by virtue, not pump as many across. Oh, that's it? Okay, cool. Anyways, question 8. Does anyone want to draw it or explain it? Let's draw it. Let's have some fun with it. Yeah. Do you want to? Huh? You want to draw it? You want to draw it like right here, or you want me to move? Go ahead. You can take one for the team. Somebody uncapped it earlier. That was not me. So
I'm just going off of what I remember it looking like, where it basically is just.
You want to draw it on my computer? Oh my god. It's gonna be really hard, but I think it'll be funny as well. So just use I think if I like draw right here, it'll make a new page. Yeah. So just draw right here. This will be like a little fun experiment. If your hand-eye coordination is good. I'm glad I'll tell my Chinese answer.
No.
No, it wasn't like a. Never mind. Bad joke. Sorry.
See, this is just how my mind like sort of remembers it. Because like it's basically moving through the gradient with counterclockwise and clockwise spin.
Wait, how about you start by drawing the attractant and where the
Oh.
vector is?
Okay, that's yeah. That's where my mind kind of blanks it because I just remember zigzags. I don't like remember how it starts like looking wise.
Okay, how about I say that?
Yeah, sorry about that.
Dude, don't say sorry. You're good. Okay, let's say this is the attractant. And then this is the bacterium. And then you don't have to draw that again every time, but how would it get to that? And I'll slow down.
So I'm assuming this is a flagellum or whatnot.
You don't have to draw the flagellum, just
just
draw where it goes.
Well, let's see. All I know is that it's trying to attract. It just will try going towards it, and then like
Wait, what did you say? It would do what?
it would try going like towards the gradient, moving through it. But if I recall, it more or less like won't hit its mark exactly, but it'll keep like spinning until it gets there, because it is still attracted. It's just trying to reach it. I don't.
So at a very fundamental level, yeah, it will not directly go towards it, and it has no way of having like any intention when you say like it'll try to go towards it. That's not how I would describe it. Does anyone else want to like take a stab at it? Gotta draw it. I'm committed to the bit. You want to keep going or you want to?
I'll leave it for someone else.
I mean, like, no, at the very root, you got it correctly, but I think there's some points to be gained. Anyone else want to? I'm gonna sit here in a side fashion. So someone else does.
You should have taken it. Oh, I can't do all.
It spins like
Bro, just
Do something. We draw already.
All right.
hands greasy. I'm kidding. Calm down.
Okay, don't fucking draw my laptop.
Do you want to use my laptop or do you want to use the phone? You can do that as well if you want.
Okay, I'll just draw on this. Oh wait, I'll just.
Dude, there's a chalkboard over there. Just go.
Got it. So I guess this is the thing. So I think this thing like rotates clockwise.
No. You're over. Don't talk about the flagellal mechanism. Just talk about the direction the path.
Yeah, okay, so I just think I'll just go right there and it'll be like a hurricane kind of thing, like swirling it.
No. Out of scope.
Let someone else take it.
Okay, I'm heavily hinting in the question, biased random walk. While that is true, whenever it's moving in a counterclockwise fashion, it's going to move forward, right? But there's something else I'm hinting at, because obviously, like, since counterclockwise is forward and clockwise is a different thing called tumbling, just like going in random directions. How does a bacteria integrate these methods into getting to its target? Because it uses both.
Go up there,
Which one you want? You want this one or you want this one?
And doesn't it like, okay, it goes in this direction, but
Well,
then-
why that direction?
because it's like, but like. So like,
Cross my colors.
because it like has to like slowly, it can't like go straight, so it has to like start making its way, but then it stops and it like suspends.
But why that direction?
The gradient. Yeah.
So,
And it stops.
you drawing a route right now? So,
And then it suspends, but like, yeah, and then it just like floats and then reach, get in the right direction, and then it goes forward. Floating
Not
in
the right
space,
direction.
like, kind of, okay.
Because, well, there is a right direction. Anything that gets it closer to the attractant is the right direction. But what I'm looking for is, the reason why I asked, like, why it chooses that direction is because it doesn't choose at all. It tumbles randomly and then moves regardless of where it tumbles. Like, let's say if it was moving backwards, Oh, he looks Chinese, sorry. Sorry, anyways, sorry, bad joke. So let's say this is like the big factor, right? They could go in this direction, but the point is that it's tumbling randomly whenever it's doing its clockwise flagellar motion. Paratricious is pronunciation. That was the first time I've heard it. But it tumbles randomly. It could go in this direction, or it could go in this direction, or this way. But the point is, wherever it ends up, not decides, wherever it ends up tumbling, the direction, or I mean, the length that it travels is kind of determined by like, no. If it's going towards the attractant, it determines how far it goes. So if it's going away, it's going to travel, let's say like 3 units, some arbitrary unit. But if it's going kind of like in this direction, like this vector right here is moving towards it. So it'll maybe be like 6. And then if it lucks out and goes straight towards it, this will be like 9. Does that make sense? So it's just like it'll travel further whenever it's heading in that direction. And somebody mentioned on Tuesday that it was because it's picking up more hydrogens as it's going towards the thing, towards the attractant. So like more hydrogens are being used to power the flagella. I don't know if that's true because the attractant doesn't necessarily have to be hydrogen. It could be like glucose.
So aside from that bit about ingredients going up. Well, it's gonna say describe how bacteria visualizes diaphragm and also travel towards an attractant for concentration. So really the concentration period doesn't matter.
No, it does. It does matter because as you're moving out from this attractant, let's say this is like 10 units of glucose, right? As it goes further away by like laws of diffusion and stuff. Okay. I'm not. So let's say it's 8, 6, 4 units per given surface area, right? Whenever it's moving from 4 to 6, glucose concentration is going to be like, dummy, I'm going to go this way. And then let's say you move from 6 to 4, it's going to be that's my understanding of it. So it doesn't necessarily have to be hydrogen concentration, unless he said that in class and somebody remembers that. So
I was thinking.
Would it be some? Sorry. Okay, so it'd be similar to the metaphor of like foggy boat on the water, lighthouse, and eventually to the point where you start noticing it gets brighter. It's not like sensing it; you're still tumbling around, but there is that part where you're starting to see the light.
well, the thing is with the lighthouse, that's like a fixed location. So once you see it, it'll be like once you see the lighthouse, you're just gonna go towards it, right? A better metaphor I would use, or analogy or whatever, would be you're blind, but you're at ACL and you want to get to the music. So you're just traveling in random directions, and if it's getting quieter, you're gonna want to make a U-turn and go in the other direction. And you're just constantly changing directions and walking until you're at the front. Well, if you're at the wrong place, you're gonna keep spinning.
So I thought he said that like it tumbles randomly, but whenever it feels like it'll know it's like a stronger gradient, it'll start rotating then. But if it goes away, then it'll tumble again. Like I thought it had that sense when he was explaining it.
I don't. Is that true? Did you say that?
In the slides, it says like in the absence of attractant, then it. But if there is an attractant, then it kind of like tests the water the other way. If not, then it it kind of like
I mean, that's the whole concept of traveling less. Because when you're traveling less in like a forward direction, you're just like tumbling more by virtue. So that's what I'm trying to do. So essentially, if you're going in the wrong direction, you're gonna tumble more just because you're traveling further less. Like think of it like a balancing out. Like this is tumbling, this is swimming. If you're closer, you're gonna want to be swimming. If you're further away, you're gonna want. Cool. How much time do we have? 8 minutes. Do we have any questions on the first or second exam that I should go over? Did everyone sign in, by the way? I just want to keep asking because I'm a numbers guy. Sign in if you haven't, but if you have, Yeah.
I saw you. Not
So it used to be 26 multiple choice and then like 5-ish free response. Yeah. I don't know if he'll keep that same format because he's changing a few things. Like
Did you
he's
say 25? I think he said 25.
oh he said 25 and 5. Yeah. And then usually like there'll be bonus points too. Like what does that add up? 25, 75? No, that's exactly 100. So
Well, he hadn't written it yet. He said this 3 responses are 5.
you said what?
Mhm.
Yeah. No, yeah, when I wrote the thing at the top, this was like exactly what my old exam said, except this was 1 through 26 and that was 27 through 32. So 3 points, 5 points. Huh? Absolutely not. No, 'cause I want to graduate. Huh?
Was it like really close?
No, I made these myself. They're just like they're harder. They're over the same concept or the concepts that you need to know.
He's not tricky with his wording. It's just straightforward, isn't it? Or would he be like, oh, it
I
doesn't
mean,
chop wood?
It'll vary. Like there will be very cut and dry questions and there will be some that are trickyly worded. And if you don't know the concept well, it also makes it tricky. So it's just like just know what you gotta know and be careful. That's really all I can tell you. Because if I say he's tricky, what are you gonna do with that information?
You're gonna
What's up?
Would you recommend like a certain chapter?
I can't because Eagly taught this part and she went over different concepts first, so I couldn't tell you what he's doing. You never heard them for us. Thank you. Thank you for coming. Good luck. No, it's totally fine. Bye. Okay. So,
Are you talking about snowdrop acronym?
a what?
Snowdrop acronym? Something about an acronym? I heard snow, an acronym.
Snowdrop. Oh, Snowdrop is something for the MCAT.
Oh, never mind. Okay.
Yeah, don't worry.
All right. I thought I like missed acronym. I was like,
No, don't worry about it. You're good.
good.